Why is it then that lions don't all have the really dark fur that you see on a certain cartoon, I guess, movie, whatever, about lions? It's because of the heat. Dark fur absorbs more radiation from the sun. So heat stroke is a real danger. So yeah, okay. Better performance? Guys, what if it just falls apart? Yeah, that's a problem, right? Okay. So the rate of enzyme catalyzed reactions will increase with temperature until the enzyme starts to denature. Why is this? Okay, so we're just basically, under the kinetic molecular theory, ramming particles together repeatedly until something clicks, right? So we know that their electron shells, their valence shells, will repel one another. Okay. So if we don't mash them together hard enough, they just bounce off each other like bumper cars. Yeah, that's pretty much what happens. So what happens when we do mash them together hard enough? Assuming that they're in the right orientation for a reaction to occur, boom, a reaction occurs. And at higher temperatures, they mash into each other faster and more frequently because, well, they're more or less held in place. So instead of fly off into distance, and later on maybe bounce back again, we're basically chaining them in place and smash, smash, smash. So what happens when we have lots of positive or negative residues, so acidic or basic residues, depending on circumstances? We're going to end up with different activity levels at different pH values. So when it's really acidic, well, you can tell that all of the normally basic amino acids, side chains, they're going to be protonated. They're going to be positively charged. This can affect their chemical behavior. And what happens to the normally acidic ones? Well, they're going to be neutral. This also affects their behavior as compared to, say, oh, at near neutral pH, the positive ones are positive, the negative ones are negative. So which ones are negative again? Well, we know that the acidic ones, carboxylic acids, they release protons to give you COO minus. So it's the acidic ones that are negative at higher pH values, and the basic ones are positive at lower pH values. Now, why is it that trypsin is more active at more basic pHs than the typical enzyme? It's because the duodenum, where trypsin is active, and the small intestine for that matter, is mildly basic. The pancreas pumps out enough bicarbonate to neutralize the chyme. All right. Why is it then that most enzymes, now that we're not looking at it, are more active at pH around neutral? It's because, well, hey, all our basic side chains are positive and all our acidic ones are negative. I wonder what this allows us to do. Oh, hey guys, ionic attractions, right? So, that's a good toolkit, isn't it? Well, yeah, okay. It's pretty obviously supposed to be. Now, for applications of enzymes, when it comes to use of enzymes in animal feed, yeah, we could think about, say, silage, which is what happens when, guys, we're cutting hay, but instead of letting it dry, we pile it in a silo so that it ferments for a while. That lets microorganisms get a good first crack at the cellulose instead of relying on the animal's digestive system, so whether it be foregut fermentation or hindgut, to deal with it. So sure, okay, we can use that as an example for use of enzymes for animal feed. What else can we do? Most humans, as adults, are incapable of digesting lactose. Lactose tolerance is actually a trait that evolved after pastoralism. Yes, okay, young mammals can all eat lactose just fine. They can digest it. But eventually the lactase production falls off in most mammals. There was a problem with cartoon depictions of cats lapping up milk. And that is because if you feed an adult cat milk, one of the more probable outcomes you'll get if you feed it a significant amount of milk is having to clean up after it. And hopefully this does not happen on carpet. All right? I think that's clear enough what I'm saying. Okay. So, what am I getting at? Oh, hey, animal feed. Hey guys, humans are animals. Cheeses and yogurts are both examples of using enzymes for animal feed. So it's because of the bacteria, generally lactobacillus, that breaks down the lactose. It also synthesizes more proteins and so on, but that's another thing. That allows us to, as adult humans, all be able to eat yogurt and cheese without ill effect. Yeah, okay, a lot of us, like me, are lactose tolerant. At least I'm lactose tolerant to about on the order of 1.5, 1.7 liters, according to a personal experiment. But there are plenty who aren't. So that's an important example. For brewing, brewing can actually also count as animal feed. Why? Well, okay, making beer, removal of sugars in light beer. Did you guys know that in the age of wooden ships and iron men, as the age of sail is sometimes called, in those days, the Royal Navy's daily ration included 2 gallons, as in nearly 8 liters of small beer? The reason was that it simply was not practical to keep fresh water safe to drink at sea. And therefore, beer was the best option available. Now, dairy, yeah, okay, that's another example of animal feed, but then again, I'm guessing the text, which you can find this table in, doesn't so much lump humans in under animals, even though we are. Okay, so we can also use enzymes as detergents. So some enzymes can be put into the powder or liquid detergents, and this can be good for breaking down of starches and fatty stains. This can also be used for softening of cotton garments, color brightening, and so on. Enzymes can also be used in the treatment of leather for many, many different purposes. So starch, well, it can be used for production of various generally less than healthy syrups, such as high fructose corn syrup. And then there's wines and juices. Yeah, okay, the proteins that come in juices, yeah, they're good nutrient content. Great, they're made of amino acids, just like all the other proteins. Fine. But most people, when they look at juice and find it cloudy, as long as it's not, say, orange juice, which if it's not cloudy, you're doing something very, very wrong, and it's probably never been anywhere near an orange. Most juices, like apple juice or grape juice, most people think of those as clear juices. So when they're cloudy, their first reaction is probably, okay, has this gone bad? As opposed to orange juice, where if it's not cloudy, well, it's never been anywhere near an orange. So you can use enzymes for, say, degrading proteins. You can use enzymes for digesting nucleic acids, although that's more used for, say, experiments. And thus, they can be used for all sorts of applications in even everyday life, really. Like, say, we need to clean this countertop. What are we going to use? A lot of really aggressive scrubbing? Or shall we just apply some nice biological agent? Okay. In summary, an enzyme is a biological catalyst. It has a specific three-dimensional shape, which is necessary for function. The active site of an enzyme is specific to a particular substrate or particular substrates. Now, yes, it tends to often be specific to part of a substrate, which is why certain receptors in the brain do not do well when treated with morphine or opium for that matter, which tend to interfere with natural endorphin production and thus produce addiction by means of, hey, no more injections or smoking equals depression in the brain, pretty much, on a chemical level. So not enough endorphins or other chemicals, as the case may be. Okay, so enzyme activity, that's affected by the substrate and enzyme concentrations. Okay, yeah, it's pretty obvious. Oh, we don't have enough to react with. Ooh, what happens? No reaction, of course. Or not enough reaction compared to the amount of enzymes that's there. Or, ooh, we just don't have enough enzyme to process all this stuff fast enough. What happens? Well, of course, we're going to end up just being near the max rate of the enzyme, as opposed to, ooh, all the substrate magically turns into product faster than the enzyme could handle. No, of course not. Okay, so then there's temperature. Oh, hey guys, if we heat this up too much, the enzyme basically flies apart. So it shakes itself apart because of all the kinetic energy. That's obviously not very good for function, right? Because it loses its specific shape. And then there's the pH. Okay, guys, instead of having these ionic bonds hold these parts of the enzyme in place, we're just going to disrupt them, and the enzyme is going to break up. Well, not break up so much, because the backbone, yeah, that takes a bit of effort to pry apart. But the pieces that we need to, say, all bite together to form the active site are no longer there. Ooh, what happens? The active site doesn't work. Okay. Competitive inhibitors enter the active site and block the binding of substrate. They compete with the substrate. What does this mean? This means that if you put in way too much substrate, you will still saturate the enzyme, and the reaction rate will be still near the maximum. Now, what happens when you have non-competitive inhibitors? Well, you can't just swamp the enzyme with substrate to crowd out the inhibitors. The enzyme just doesn't work as well as it would if there were no inhibitors. That's presuming that the inhibition is reversible. If the inhibition is irreversible and you add enough inhibitor, well, it'll be generally a lot more obvious. This also kind of depends on whether or not binding the substrate affects the structure of the enzyme at the inhibition site. Why? Oh, hey guys, the enzyme is constantly handling substrate. Ooh, the inhibition site is not open in this conformation. How is the inhibitor supposed to get in there? Not nearly as effectively, right? Of course, this kind of depends on when you're adding the inhibitor, if it's irreversible, or if it is reversible, how much substrate there is versus how much inhibitor there is, and how much enzyme there is. So this is something that we can contemplate a little bit mentally when it comes to, oh, what about more complicated circumstances than just, okay, inhibitor in, enzyme change shape, substrate doesn't bind anymore. And then, of course, there's the activation. So allosteric activation is the counterpart to allosteric inhibition. So non-competitive inhibitors and uncompetitive inhibitors, which are actually separate categories, are generally both considered allosteric, and they inhibit or stimulate enzyme activity by altering the affinity of the active site for the substrate. Now, given that this here, we're talking about allosteric regulation, hey, what about activators that aren't allosteric? I mean, we know that there are competitive inhibitors and non-competitive inhibitors, so allosteric inhibitors. Hey, we know that there are allosteric activators. What about activators that work at the active site? Generally, they are considered coenzymes. Hence, we don't use that term much. So there's no competitive activator, because first of all, it doesn't actually compete for the active site, and well, we just call them coenzymes anyway. Okay, so biochemical pathways, they often use feedback inhibition, which is a form of negative feedback. So in other words, the textbooks, the textbook we are basing this course off of uses negative feedback inhibition, but this is a little bit of a Department of Redundancy department thing. That is a trope, as in a commonly used plot device in fiction, by the way. So, negative feedback is a matter of, okay guys, there's been a change. Stop. Far enough. Done. Or, whoa, too far. Go back the other way. Positive feedback is more of the fight variety. That's basically egging the process on. And it's very rare in biology other than relatively one-time processes such as childbirth. So negative feedback is, okay guys, there's been a change. We've fixed something. Done. Or, oh, something's off. We need to fix it. Okay, we fixed it. Done. If you want the whole process. So when it comes to regulating enzyme pathways, so biochemical pathways, you generally go, hey, there's enough product. Stop. So how do we do this? Well, we need to make the product inhibit an enzyme at or very close to the start of the pathway, or at least at the branch of the pathway that's closest to this particular endpoint. Because who knows, maybe the other branches in the pathway, or set of pathways, is actually useful right now. Okay, so there are many industrial and commercial uses of enzymes, both in vivo, so in life, and in vitro, in glass. Why? Guys, lactobacillus used for fermenting yogurt is alive. And what about making cheeses? Well, in addition to bacteria, there's also fungi potentially in there, as is in the case for, say, cheeses that have blue streaks or black spores or whatever inside. Those are fungal spores. Now you might be thinking, whoa, why would a fungus ever put spores, which are used for reproduction, inside a piece of cheese? Like in the air spaces in there even. Or wait a second, whoa, I've eaten that sort of cheese. It's not even necessarily exposed to air spaces in there. Well, the thing is, you take the cheese curds, you lay them out, you let them pretty much rot for a bit, and then you take them, collect them up, and you compress them into shape. So, what is now inside the cheese was outside at one point or another. That's why the fungus put out spores out there. Okay, so what about if we don't use life forms? Well, we still have some detergents that use some enzymes. We have various other applications of enzymes, such as, oh, hey guys, well, suppose we need to do some sort of experiment. We are not going to be necessarily using living things. If we say just, oh, I have this sample here. I want to digest all the DNA in this sample, or I want to digest all the RNA in this sample. Or, well, I want to digest all the proteins in this sample. What do? Of course, we add some sort of enzyme extract, be that DNase. Spelled like this. We know that lactase digests lactose. So enzymes are named with the ase ending. The reason why DNase is generally abbreviated like this is because it's just convenient. And then of course there's RNase and then protease. RNase and protease. Okay, so now that we know that enzymes help make reactions go much faster than they would otherwise by holding the components in approximately the right conformation and letting them basically clack, 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 clack together until they finally click. What happens next? Well, that's it for this section. It's also it for this chapter. Next chapter, we start talking about some of the emergent properties of biology. After all, hey, if we have just a pile of components, that's not going to let you outrace some other guy who's on foot. Whereas if you have a bicycle that's made out of those same components, yeah, you're going to outrace him. Now, I'm not counting the assembly time, because if the race is long enough, yeah, okay, you could outrun a guy on foot if you, even if you have to assemble the bike first. Do not play smart like that. Okay, so wait a second. If we talked about the basic molecular components of life, what's the next step up? Well, okay, it's easily argued that it can be organelles, such as, say, chloroplasts and mitochondria. And, well, the next section is about the inner workings of cells, which, of course, talks a great deal about the organelles. So, yes, that claim that the next section should be about organelles is partly correct. So we'll see each other again next section.